Okay, so a pleasant morning to all of us. Kumusta kayo lahat dyan from all points of the country? So we would like to thank you all for responding to our invitation to this webinar entitled Concept Enrichment on Cellular Respiration. This is an online forum brought to you by Project STAR, or the Science Teacher Academy for the Regions of the Science Education Institute, Department of Science and Technology. So I understand you were sent materials for your advanced reading, so you may have a complete grasp of the topic to be discussed today. Yes, there are a total of 67. I see here 67 participants. Of course, mostly science teachers, preferably teaching in the junior and senior high school. Okay, who have signed up, and I wish that you find this forum valuable to you as a professional and hope that learnings may be applied to your schools or workplace when you return to school after this pandemic, according to Secretary Briones sa August 24, tama ba? So before I turn you over to our resource person, may I request participants to turn off their video so we can focus on the presentation. I understand your microphone will be automatically muted. So, but later on, it may be turned on during the open forum if there's a need. Okay. So now, we have invited a resource person to share with us this very interesting topic. She is a faculty member of the St. Louis University, but is currently on leave to pursue her PhD in botany at the UP Los Baños. She is a star trainer from the Cordillera Autonomous Region, or CAR, and has been with us for around 4 years already. So folks, it is a great pleasure to introduce to you our resource person, Miss Majal Rani Espiritu. Take it away, Ma'am Maj.
So good morning, teachers. Welcome to the Content Enrichment webinar on bioenergetics, specifically on cellular respiration. To start with, let us look at an overview of cellular respiration. Cellular respiration occurs in all living forms, be it the simplest bacteria to the most complex plants or animals. Cellular respiration is important since this is the process that forms ATP, the major molecule used as energy source, and the products of cellular respiration are used in building new molecules for cells and body repair and for growth and development. So in the diagram, we would see the reaction of cellular respiration. That would be C6H12O6, which is a large molecule together with oxygen can be broken down into simpler molecules, which would be carbon dioxide and water. So this breakdown of large molecules into simpler ones is called a catabolic process. Thus, simply put, we have cellular respiration as a catabolic process. Now let us focus on the mitochondrion, the main organelle for cellular respiration. And I hope that most of us already understand the structure of the mitochondrion and its function as the major powerhouse of the cell. So the mitochondrion looks very similar to a prokaryotic cell or a bacteria, and its functions would actually involve chemosynthesis aside from many others. So let us review its membranes and compartments because these are the ones relevant to our discussion. The membranes would be the outer membrane, seen here as purple, and we have the inner membrane, seen here as pink. So the outer membrane is smooth and permeable. It is made up of 50% proteins, which is mostly porins. Porins, teachers, are water passages. Now the second, the inner membrane, would be folded. As we could see, these are cristae. And this would be the site of the electron transport chain and chemiosmosis. This is an impermeable partition composed of 75% protein and cardiolipins, which makes the inner membrane impermeable, and likewise the transport proteins, which we would be mentioning a little later on. Now for the compartments, we would see the matrix and the intermembrane space. Now the intermembrane space would be found between the outer and the inner membrane, and the contents of which would actually be similar to that of the cytosol. The inner compartment, however, which is found inside the inner membrane, is the site of the Krebs cycle. Now, for the processes of cellular respiration, we have three main parts. However, we would have to add to it the last and the final step of the cellular respiration, which is CHEMIOSMOSIS. Now, for the entire process, you can see glycolysis to happen in the cytoplasm of the cell. And then the molecules would be entering the mitochondria inside the matrix. That is where Krebs cycle happens. And as you can see in the inner membrane, this is where the electron transport chain and the chemiosmosis happens. Before moving on to the specific steps, let us first discuss the ATP-ADP cycle. Now, gagawin lang po nating simple ito para hindi po tayo mahirapan. Bahala na po tayo kung paano natin i-explain sa mga estudyante natin. Pero basically, the ADP-ATP cycle involves the transfer of phosphates. So here we would see the symbolism to be PI, or that is inorganic phosphate. Or sometimes, ang ginagamit natin ay P, excuse me. Or mas mahirap po kasi naggamitin natin is PO3, negative 2 charge. So kadalasan sa school ang ginagamit po natin ay P. Now these phosphates are part of ATP or adenosine triphosphate. ATP is the molecular unit of currency in cell energy transfer. This means that ATP is the molecule used whenever energy is needed for a process to occur. One or two inorganic phosphates are traded. Ipinapalit po siya in order for energy to be produced. ATP is made up of, if we look at the diagram below, ito po, we have adenine as the nucleotide. We are familiar with the nucleotides. We have A, T, G, and C. Dito ang ginagamit po natin ay si A, si adenine. And then we would have the sugar ribose. It is a hexose. If we combine adenine and the ribose, we come up with the term adenosine. And then we would add the 3 phosphates, thus we have adenosine triphosphate. Now, please focus on the blue box. We observe here the removal of P from ATP. Lalabas po si P. This is called as dephosphorylation. Now, if we remove 1 P from ATP, it results to ADP, that is adenosine diphosphate. While ATP, you remove 2 phosphates, you come up with AMP, or adenosine monophosphate. You would have the prefixes there to indicate the number of phosphates present. Now, as we can see, in dephosphorylation, energy is released. Lumalabas po ang energy. So, ito po ay nagagamit po ng cell or nagagamit dun sa reaction ng molecule. Now, in ATP-ADP cycle, we have another. Please focus on the blue box again. Here, it is different. We add a phosphate. So, ADP, we add 1 phosphate, we come up with ATP. Thus, if you would see there, you have energy to be absorbed, indicated by the yellow arrow. Okay. So, ATP to ADP, dephosphorylation. ADP to ATP, phosphorylation. Okay, this would be the different types of phosphorylation. Bakit natin babanggitin ito? Ito po ang pinaka-heart ng ating cellular respiration. So the first one is substrate level phosphorylation. What is it? So here we have a substance to work on, this in orange box. We call this as the substrate. And then this is acted upon by the enzyme. This large purple, I hope you can see what I'm pointing at. Large purple structure here. Tapos meron po siyang dalang ADP. Now the enzyme removes the P from this substrate and adds it to adenosine. So nag-form po tayo ng ATP. And guess what? You have energy to already be absorbed and found within ATP. Okay? Now this process is undergone in glycolysis and Krebs cycle. The second process is oxidative phosphorylation. Malayong mas mahaba po ang drama nito. Here we see a glucose molecule and an oxygen molecule which would undergo a series of processes, which is why we have lots of arrows here, and then eventually bring out carbon dioxide, water, and of course, ATP. Now, this oxidative phosphorylation is the main process which is undergone in ETC or electron transport chain and likewise chemiosmosis. Okay, let's start with glycolysis. As an overview, glycolysis has two phases. A preparatory phase, which is the investment phase. This is made up of 5 steps. Now, please see the green circle over here. We have to use 2 ATPs. Okay, which is why we invest 2 ATPs. Gagamitin muna natin siya. Bago po tayo kumita ng ATP dito sa payoff phase. Okay? Please observe that towards the end of this phase, we result to a 3-carbon molecule plus a phosphate. Ganon din po yung isa, 3-carbon molecule plus a phosphate. Now, the second phase would be the payoff phase. Here, we would be making use of NAD, which is in blue circle, and ADP. Okay? However, dalawa na sila. Tig-dalawa. Kasi nag-branch off po tayo. So from NAD, we form NADH. And from ADP, we phosphorylate that to form ATP. Okay? The end of that would be a 3-carbon molecule without the phosphate. This is what we call as pyruvate. Okay, so let's go to the details. The first enzyme of the investment phase, so 5 steps po ito, would be a hexokinase. So naka-highlight po ang kinase. Ano po siya? Ang gamit po ng kinase ay nagtatransfer ng phosphate. Okay? So we would see that from glucose, we come up with glucose 6-phosphate. How is that so? ATP gave up one of its phosphates and became ADP. Saan nag-attach si phosphate? Count po natin dito at the upper right-hand corner, we have the positions 1, 2, 3, 4, 5, 6. Compare that to the resulting molecule down below, dun po nag-attach ang ating phosphate. Kita po natin yon. Okay. Next one. We have phosphoglucose isomerase. Now again, we have the term isomerase. So it would be transforming it in another version of a molecule. Kaparehong-pareho din po ng molecular formula niya, pero ibang-iba po ang structure niya. So we have glucose 6-phosphate to be transformed into fructose 6-phosphate. Anong pinagkaiba? Ito lang pong fructose. Bakit? Si glucose at si fructose ay mag-isomer. So ito po si glucose, ito na po si fructose. Pansinin po yung difference ng itsura nila. Nagpalit po ba ang position ng phosphate natin? Hindi. Nandun pa rin po siya sa position 6. Next, we have the third. The third step is phosphofructokinase. Again, kinase ulit ito. So, nag-split po tayo sa first at saka sa third steps. So, meron po tayong ATP ulit na ginamit dito. Tinanggal natin ng P para siya ay maging ADP. Now, look at this. you would have the other P to more or less attach in the first position. So down below would be fructose 1,6-bi. Dalawa na po ang phosphate natin, biphosphate. Now, if we stretch this out, this would be the resulting molecule, fructose 1,6-biphosphate. Pero kung titignan natin, the phosphates would be attached to either end already. Next, fourth one. The fourth step involves aldolase. Here we would have a split of the 6-carbon molecule. So magkakaroon po tayo ng split. Okay? Kaya ang result po natin ay 3 carbon molecules. That's 1, 2, 3. The other is also 1, 2, 3. However, magkaiba ng itsura ang dalawang molecules na to. We have the first one to be dihydroxyacetone phosphate and the other to be glyceraldehyde 3-phosphate. These are isomers also. Pareho po sila ng chemical formula pero magkaiba po sila ng structure. Sa isa, naka-attach ang phosphate natin sa first carbon. Sa isa, naka-attach ang phosphate natin dun sa third carbon. Kaya po siya glyceraldehyde 3-phosphate. Anong pinagkaiba? This dihydroxyacetone phosphate actually is transformed into triglycerides. And then eventually, magiging fatty acids siya or fats siya, excuse me. And then, ito po ay isostore na ng body. Ang glyceraldehyde 3-phosphate, on the other hand, is readily transformable so that this could actually move into the other steps of glycolysis. Okay? So, kung titignan natin, magkaiba sila, pero parehong-pareho sila, kaya isomers po sila. So we have the fifth one, TRIOSE phosphate isomerase. Again, isomerase, magta-transform po tayo sa isomer. Ngayon, etong dalawang isomers, yung sinabi po natin kanina, they could either interchange or change into the other. So D-hap could become G3P or G3P can become D-hap. Kung marami po tayong kinain, ang G3P magiging D-hap, masostore na fats. Kung hindi ka kumain at kailangan mo ng energy, yung D-hap po ay yung fats mako-convert into D-hap and babalik po ito sa pagiging G3P tapos mag-undergo ulit ng glycolysis. Sixth, we have glyceraldehyde phosphate dehydrogenase. From the term D, we remove or we transfer and hydrogen. So we remove or transfer a hydrogen. So this is the first phase by which we already make use of the NAD. Okay? Now take note that ang G3P natin, pagka pre-process natin ito, laging tigda-dalawang molecules na from stage 6 or phase 6. Bakit? Kasi po, nag-split po tayo sa fourth step, nag-transform tayo sa fifth step, sa sixth step po, meron na po tayong process na i-undergo ulit. So that would be NAD, tapos meron po tayong available na hydrogen, if we would be looking at this. So we have G3P to have a hydrogen. This would be moved into NAD, forming a NADH. May papalit po sa ating position. It would be a phosphate. Where did we get this phosphate? Phosphates would actually be free-floating in the cytoplasm. These are ions that are free-floating in the cytoplasm. So they could attach easily to this molecule. So we already have 2 phosphates in this particular molecule. Thus, we have the term 1,3-biphosphoglycerate. Seventh, the seventh step would involve phosphoglycerate kinase. Again, kinase siya. Magta-transfer po ulit tayo ng P. Now, take note that unlike the investment phase, The P group here comes from the molecule itself, not from ATP. So the donating molecule would be 1,3-biphosphoglycerate. I-donate po niya ang isang phosphate niya. Okay? So pag dinonate niya ang isang phosphate niya, si ADP magiging ATP. Tapos, iisa na lang po ang natira nating phosphate. Thus, we already have that to be 3-phosphoglycerate. Now we have the 8th step. 8th step is phosphoglyceromutase. Mutase would be resulting to a translocation only. So this would actually be from the 3rd position of the phosphate, it transfers into the 2nd position, which is why here in the resulting molecule, instead of the position 3, we have the position 2. Thus, we have 2-phosphoglycerate. Again, we form 2 molecules of this. Second to the last step would be enolase. Enolase removes water. So we have the H coming from this position and the hydroxide coming from this position. Okay? So pag nag-combine yan, lalabas na po siya as water. And the resulting molecule would be phosphoenol pyruvate. Again, 2 molecules of this. Now the last one would be pyruvate kinase. Again, kinase siya. So you would have to remove this phosphate from phosphoenol pyruvate, add that to ADP, resulting to ATP. So the resulting molecule would be pyruvate. Again, we have 2 molecules of this. Now let's have a review and let's try to compute the gained and used ATP we have. So in the investment phase, we made use of 2 ATPs that are utilized. No other energy or products are produced. In the payoff phase, we made use of 2 NAD molecules to form this into NADH. And then we made use of 4 ADPs to turn this into 4 ATPs. Okay? NADH would proceed to ETC. Wala pong ibang pupuntahan si NADH kundi sa electron transport chain. Likewise, we have the formation of water. Okay. Now, if pyruvate is already available, it may take 2 paths. The first path is when it is left in the cytoplasm where you do not have oxygen, then it undergoes anaerobic respiration or fermentation. There are 2 types of fermentation. We have lactic acid fermentation and we have alcohol and carbon dioxide or alcohol fermentation. So in lactic acid fermentation, this occurs in our muscles. We usually experience the buildup of lactic acid as cramping. Cramping happens when there is excessive physical activity, pero wala po tayong intake masyado ng oxygen. So it's best that when you exercise, you couple it with deep breathing. So in lactic acid fermentation, we only have 1 enzyme, lactate dehydrogenase. So again, dehydrogenase, it removes or transfers hydrogen. Pero ang pagkukunan natin ay hindi po ang molecule, ang pagkukunan natin ay si NADH. So si NADH po pwedeng siya yung galing sa glycolysis natin. Pero po pwedeng itong NADH ay available din na papunta sana ng ETC pero ninakaw po muna ng lactate dehydrogenase. So NADH is broken down and releases its hydrogen to be added into the position where you have the 2nd carbon of pyruvate. Thus, you would result into lactate or lactic acid. Tapos meron na po tayong i-release na NAD. In alcohol fermentation, this would be of 2 steps, unlike the first one, and this is usually attained with the help of microorganisms like yeast and bacteria. This process is best exemplified in beer or ethanol production. Now, the first step would be involving pyruvate decarboxylase. So decarboxylase removes carbon and oxygen in the form of CO2. So here we would see in pyruvate the position of CO2 with the action of pyruvate decarboxylase. Ilalabas po niya ang CO2. Ang isang action din po ni pyruvate decarboxylase, nag-attract po siya ng isang hydrogen at inattach po niya sa pinag-alisan ni carbon dioxide. So the resulting molecule is acetaldehyde. Okay? The next step is alcohol dehydrogenase. From the term dehydrogenase, again, we remove or transfer hydrogen. So here we would see the use of NAD. Okay? Tinanggal si NAD, kumuha din ng isang hydrogen, tapos ang hydrogen na ito at yung hydrogen galing sa NAD, dinagdag po sa acetaldehyde. So the resulting molecule is ethanol. NAD would again be released. Now, if pyruvate enters the mitochondria, where you would have the presence of oxygen, then here we would have the aerobic pathway to be followed, or aerobic respiration. This is what happens actually na familiar tayo, aerobic respiration, kasi ito talaga ang pina-process natin. So we would have the Krebs cycle, the electron transport chain, and chemiosmosis. Now, before we go any further to the Krebs cycle, we still have to prepare pyruvate. So pyruvate would involve, the preparation of pyruvate would involve the enzyme pyruvate dehydrogenase. Now, from here on, marami pong functions ang nag-iisang enzyme. Kaya marami po ang nahihirapan sa Krebs cycle dahil marami po siyang actions. So isa-isahin po natin. From the preparation of pyruvate, pyruvate dehydrogenase would, number 1, remove carbon dioxide from pyruvate forming acetyl. Ito po ang ating carbon dioxide. Okay? Natanggal po siya. Okay? Ito po ang natira. Ito na po ang backbone ng ating acetyl. Okay. Another, we have pyruvate dehydrogenase to make use of CoAsh. CoAsh is CoAsh is coenzyme A with sulfhydryl, kaya siya CoAsh. Meron siyang H doon. Now this H would be broken down and then this would be added to NAD, forming NADH. Papunta ulit po si NADH natin dun sa ating electron transport chain. Now, eto namang coenzyme A and S natin, or S coenzyme A, this would be added into acetyl. Kaya ito po ay magiging acetyl coenzyme A. Now, we are prepared for the Krebs cycle. The Krebs cycle is also called as the citric acid cycle or TRICARBOXYLIC acid cycle. Now, as we can see, we would have citric acid to be the first, the very, very first molecule to be formed in Krebs cycle. Okay? Meron din po siyang tatlong carbon dioxide. Kaya ang tawag natin dito ay tricarboxylic acid din. So, Krebs cycle is the central metabolic pathway of the cell since it produces many intermediates. Intermediates would actually be substances that would still be used in other processes. Makikita po natin ang iba-ibang mga pangalan mamaya ng mga products natin, or sometimes hindi po products pero mga ni-release po galing sa cycle natin, and this would be going into other processes as well. Now, for each step, you please follow the blue arrow because this is directed in a cyclical manner. Thus, the arrow may appear to point to different directions. Okay, so acetyl coenzyme A, from the preparation of pyruvate, is acted upon by citrate synthase. So from the term synthase, this is in charge of synthesis or the formation of different compounds. Citrate synthase does 4 different things here. Excuse me. So number 1, acetyl coenzyme A is broken down into acetyl and S-CoA. So here we would see acetyl coenzyme A. This is the acetyl part. This is the S-CoA. Now we also have oxaloacetate. Saan po siya galing? Si oxaloacetate galing po sa last step ng Krebs cycle natin. Product po siya. So that oxaloacetate would be combined with acetyl to be forming citrate. Pero that's not actually the end. Meron pa po tayong hydrogen coming from this water molecule to be added. Okay? Which is why we form CoAsh again. And likewise, another hydrogen still from water would be added over here in order for us to form citric acid. Now, the second step involves aconitase. Aconitase is similar to an isomerase. Yun nga lang po, the difference is it is very specific to only transform citrate to isocitrate. Yun lang po ang pinaka-function niya. So, aconitase would be very specific only for the transformation of citrate to isocitrate. Yan lang po ang mangyayari sa second step. Now, the third step would involve isocitrate dehydrogenase. So, as a dehydrogenase, again, it removes or transfers hydrogen. So, the first function would be the transfer of hydrogen from isocitrate. We see here the green boxes. This would be added to NAD in order to form NADH. And we would have a COO from citrate. This would be released as carbon dioxide. Okay. Now, if we go back, isocitrate is a 6-carbon molecule. Here, we have alpha-ketoglutarate, which is a 5-carbon molecule. Saan napunta si carbon? Kasama po siya ni carbon dioxide. Lumayas na po. And we have the fourth. We have another dehydrogenase. Again, it would be removing or transferring hydrogen. It performs 4 different steps. Okay, 4 different functions. So we have CoAsh to be broken down into S-CoA and H. So CoAsh becomes CoAS and H. So you would have the H to be donated to NAD. Magfo-form na tayo ng NADH. And we also have the COO coming from alpha-ketoglutarate. I-release po niya yan in the form of CO2. Saan po nagpunta si S-CoA? Papalitan po niya yung position ni carbon dioxide sa alpha-ketoglutarate. Thus, we already have succinyl coenzyme A. Take note, yung previous slide natin, alpha-ketoglutarate, 5-carbon molecule. Succinyl coenzyme A, 4-carbon molecule. Saan ulit napunta si carbon? Sumama ulit kay carbon dioxide. Lumayas na din po. Succinyl coenzyme A, synthetase. This would be the 5th step. Synthetase is different from synthase. So that you have the link or the bond. Mamaya makita natin kung ano tong link or bond na sinasabi kong ito. Okay, so here, the first step is GDP is turned on, forming GTP. Bakit wala tayong ATP? Bakit GTP? In this particular process, we have GTP, or that is guanosine triphosphate. Pinalitan lang po natin yung adenine ng guanine. Kaya meron po tayong GTP. Now, this particular molecule is actually turned off when it is GDP. and it is turned on when it is GTP. Now, when it is turned on, it combines the free-floating phosphates and the free-floating ADPs so that it would form ATP. So, indirect po ang formation natin ng ATP dito. Kailangang ma-turn on muna si GTP para mag-combine ang P at ADP para mag-form ng ATP. So, that is the first function. The second function would be the removal of S-CoA over here, and then this would be brought out. Okay? Resulting to succinate 4-carbon molecule. Next one, we have the sixth, which involves the third dehydrogenase. So, kung titignan po natin, 2 beses na po tayo kanina nag-form ng NADH. Dito po, magpo-form naman tayo ng FADH. So, again, dehydrogenase siya. We remove or transfer hydrogen. So, the first function is to remove hydrogen. This would be the first carbon group. This is the second carbon group. This is the third carbon group. Tig-2 ang hydrogen natin dyan. Inalis po natin ang tig-1. Dinagdag natin sa FAD para maging FADH2. Nagtira po tayo ng tig-1. Thus, you would have the formation of fumarate. Okay? We have the last, second to the last step, we have fumarase. And from the term, this is in charge of the breakdown of fumarate. So, simple lang ang ginawa niya dito. Kumuha siya ng water at break down niya ang water. And it would be adding 1 hydrogen here and the other OH here, forming malate. Still, malate is a 4-carbon molecule. And the last is malate dehydrogenase. From the term dehydrogenase, again, you remove or transfer a hydrogen. Okay? So, saan galing si hydrogen? Dito po siya galing. So, malate dehydrogenase breaks down malate, gets its hydrogen, adds it to NAD to be forming NADH. And the resulting molecule is oxaloacetate. If you remember, oxaloacetate would be one of the first reactants that we would be using in order for us to form citrate. So, summary of the Krebs cycle. To summarize, the start of a single cycle involves acetyl coenzyme A. Kung 2 cycles tayo, we would make use of 2 acetyl coenzyme A. Okay? Pero ito, isang cycle lang ang pinapakita natin dito sa diagram. So, if we make use of 1 acetyl coenzyme A, this would be combined with oxaloacetate, a 4-carbon molecule. And then, pag kinombine natin sila, we result to a 6-carbon molecule here. Okay? Now, as we go through the cycle, the 6-carbon molecule becomes 5-carbon molecule, and then it becomes a 4-carbon molecule. All throughout, 4-carbon molecule na po yan hanggang sa maka-encounter ulit siya ng acetyl coenzyme A. Along the way, we would be making use of NAD. That would be 1, 2, and 3. Ang FAD, gagamit tayo isang FAD lang. So kung 2 cycles ito, we would be making use of 1, 2, 3 times 2, 6 na NAD. Tapos ang FAD natin, 1 times 2, 2 FAD. To be resulting to 1, 2, 3 times 2 na NADH. Ang FADH natin, 1 times 2. Okay? So this would be the products for the 2 cycles. We have 2 ATPs. We have 2 FADH and 6 NADH to go into the electron transport chain. And if you remember, we were able to release 4 carbon dioxide. Ito na po yung breathe out natin. Now, please take note that as we would be moving through, we would have FADH2 and NADH to be high energy molecules. Bakit? Ang bawat FADH2 would produce 2 ATPs. Ang bawat NADH would be producing 3 ATPs. So if we have 2 cycles, then FADH2 times 2 cycles times 2 ATPs ang ginagawa ng bawat FADH2 will result into 4 ATPs. Sa NADH naman, pag dalawang cycle at sa bawat NADH, meron tayong 3 ATPs, then that would be 6 ATPs for each NADH molecule. Now, before we proceed to the ETC, actually we could already calculate the ATPs that are formed from one glucose molecule. Why? Kasi we already have the products of the Krebs cycle and glycolysis na gagamitin natin sa ETC. So po pwede na natin i-compute dahil alam naman na natin that one FADH2 forms 2 ATPs and one NADH would form 3 ATPs. Thus, in glycolysis, we made use of 2 ATPs and we produce 4 ATPs. Thus, our net would be 2 ATPs. In NADH, on the other hand, produce din ito sa glycolysis. Meron na tayong dalawang paths. Remember, nag-split tayo sa 4th step. So we would have 3 ATPs for each NAD, resulting to 6 ATPs. So that would be 8 in total for glycolysis. In pyruvate preparation, meron ba tayong na-form na ATP? Wala po. Ang na-form lang po natin ay NADH. Pero sulit-sulit po tayo dito kasi ang isang NADH is 3 ATP. Dalawang paths po ito times 3 ATPs, then we have 6 ATPs. In the Krebs cycle, we have 1 ATP that was formed, if you remember, yung indirect formation nito by the turning on of the GTP, and then we have 2 cycles, so that would be 2 ATPs. Okay? Now, yung na-produce natin sa Krebs cycle na FADH2 at NADH, i-count na natin sa electron transport chain. Kasi dito naman po napaprocess talaga yung ATP. So here in the electron transport chain, syempre 2 paths pa rin po ang electron transport chain na FADH. So that would be 2 times 2, we have 4 ATPs for the single FADH2. And for the 3 NADH molecules, following the 2 paths, and we have 3 ATPs for 1 NADH molecule. That would be 3 times 2, 6 times 3, 18. Add po natin lahat ito. Our grand total is 38 ATPs. This is the ideal. Now we have the electron transport chain and of ATP synthesis, or what we have as energy production. So if the Krebs cycle is the heart of all biochemical pathways, the electron transport chain and chemiosmosis is the heart of energy production. So ATC is also known as respiratory chain. Mas maganda pong pangalan niya ito kasi sa photosynthesis, meron din po tayong electron transport chain. Pero para specified po na, excuse me, sa mitochondrion ito, then we make use of the term respiratory chain. Excuse me. Now the ETC would be made up of, we could see here, 1, 2, 3, 4 molecules which would be in Roman numerals. These are what we call as complexes. Then we have these 2 small molecules here. One is named as Q, the other is site C. This would be our prosthetic groups. And here we have the ATP synthase. Now you would see that the entire process here is actually oxidative phosphorylation. Maalala niyo yung mahabang-mahabang proseso kanina ng oxidative phosphorylation. Dito na po tayo mag-uumpisa. Pasa-pasahan lang po ito. The first part is the electron transport chain. And the last part is chemiosmosis. So we would have the list of the 4 complexes here. They would be symbolized with Roman numerals. So the first one is NAD dehydrogenase. The second is succinate dehydrogenase. So from the term itself, they again would be removing hydrogen. Okay? But the first complex removes hydrogen from NADH, forming NAD. The second removes hydrogen from FADH2, forming FAD. Okay? Now, before we go to the third, please remember or recall the redox reaction, oil rig. If you remember oil rig, oxidation is loss, OIL. Reduction is gain, RIG, oil rig. Okay? So here we have a reduction or a reductase. Reduction again is gain. So cytochrome BC1 receives or gains electrons from complex 1 and 2. Okay? Now cytochrome oxidase, which is the fourth, oxidation is loss. You remember? Okay? So it would donate electrons and give it to oxygen. Now, if you try to look at the other functions, 1, 3, and 4 would be donating hydrogen. Hydrogen would be pumped from matrix. The matrix is inside, and it pumps the hydrogen towards the intermembrane space, which is why you have 3 arrows here. Okay? So hydrogen is pumped out from 1, 3, and 4. Yun nga lang, dito sa 1, 4 ang hydrogen na pinapump out. Sa 3, 4 din. Sa 4th, 2 lang. Okay? Tignan natin yan later on. Now, this one, the second complex, bakit hindi siya nagpapump ng hydrogen? It does not really pump out hydrogen because, if you remember, succinate dehydrogenase is still part of the Krebs cycle. So, kumbaga, meron tayong Krebs cycle na naka-attach dito. Malapit na malapit lang siya. So, bawat dito sa Krebs cycle, pag nag-form ang NADH, diretso sa complex 1. Pag nag-form ang FADH2, diretso agad siya dito. So, pagpasok niya dito, yun lang po ang pinaka-function niya. Taga-remove lang po ng H from FADH2. Pero itutulak po ba niya ang H? Hindi po. Now we have the prosthetic groups as mentioned a while back. We have Q or ubiquinone and we have cytochrome C. Okay? Now the functions of these two are carriers. They would actually be moving about. Ganyan po ang movement niya. Excuse me. So, ubiquinone would be carrying 2 electrons from 1 and 2, dadalhin niya sa 3. Siya ang taga-kolekta dyan at dadalhin niya sa 3. Si cytochrome naman would be collecting or carrying electrons from 3, bringing it to 4. Okay? Now, again, you would see that there would always be 2 electrons. 2 electrons. Okay? Now, the last one, ATP synthase is in charge of chemiosmosis. That is the only enzyme which is in charge of chemiosmosis. And this would actually be the final phosphorylation that happens. So here we have ADP plus P, it results to ATP. Now take note that ATP synthase is not really positioned side by side with all of the other complexes here. Hindi po laging ganito ang position niyan. ATP synthase is actually nakakalat po yan sa inner membrane natin. All throughout nakakalat po siya. Pero just for instructional purposes, andito po tayo. Magkakatabi sila para lang po makita natin yung flow natin. So here we have oxidative phosphorylation. This is the entire process na hindi natin na-discuss kanina. Kasi ito po ang talagang nangyayari. Kunin po natin na example C, NADH. Okay? So NADH would be broken down by complex 1 into NAD plus hydrogen. Pero si hydrogen would be further separated into one proton or an H plus and then 2 electrons. Okay? So, kung ang ating NADH ay break down na ni complex 1, dito sa loob ng ating complex, meron na tayong 2 electrons. Saan po napunta si hydrogen? Andyan din po siya, nag-aabang. Anong aabangan niya? Etong complex, ipapower po ng ating 2 electrons, hihigop siya ng hydrogen galing sa matrix. 3. 3 plus 1 galing kay NADH. Ito na po ang ibubulwak niya palabas. Okay? So this is actually the pathway of NADH electrons. So 2 electrons from NADH would enter complex 1. Hydrogen, 4 hydrogen ions would be pumped out, and then the 2 electrons would be carried by ubiquinone. Ubiquinone transfers the 2 electrons to complex 3. Complex 3 pumps out 4 electrons from matrix to intermembrane space. The 2 electrons would be carried by cytochrome C. Cytochrome brings the 2 electrons to complex 4. Now, anong mangyayari dun sa 2 electrons natin dito? Now, very important to remember is that the 2 electrons here would be received by oxygen. Okay? Ang free oxygen kasi natin is in the form of O2. However, the 2 electrons would only be attaching to 1/2 of our free O2. So that would be 1 atom only of oxygen. And then immediately, kukuha siya ngayon ng hydrogen ions dito sa matrix. So 2 hydrogen ions plus yung ating O na may nag-combine na 2 electrons. And then this would be transformed into water. Kuha ba natin yon? Okay. NADH, mabreak ito into hydrogen. Hydrogen forms 1 proton and 2 electrons. So here you have 2 electrons, 1 hydrogen. Kukuha ng 3 hydrogen sa baba. Pump ang 4 palabas. Si 2 electrons, mag-move sa ubiquinone. Dadalhin ni ubiquinone sa 3. Mapapalakas ulit si 3 or ma-energize si 3. Pump si 3 ng 4 galing sa matrix, 4 hydrogen ions palabas sa ating intermembrane space. Si 2 electrons dito, bibit-bitin ni cytochrome C, dadalhin sa complex 4. Now, if you remember again, meron tayong 2 electrons dito. It combines with 1 single atom of oxygen. And that single atom of oxygen is the receptor and then attracts 2 hydrogen ions, combines it together to form water. Now, bakit nga ba 2 lang ang ipapump ng complex na ito papunta sa intermembrane space? Kasi, di ba, kailangan natin ng 2 dito. So, yung hinugot niyang 3 galing dito, ito ngayon ay ilalabas niya. 2 lang dito. Yung 2 gagamitin niya dyan. Nakuha po ba? Now, pagka nagkakaroon po kunwari ng cyanide poisoning, cyanide would be targeting complex 4. In plants, it would be rotenone. It targets complex 4. So anong nangyayari? Pagka natatarget ang complex 4, hindi po niya tinatanggap ang electrons. Or kung tatanggapin po niya ang electrons, hindi po ito nagko-combine with oxygen. So, mamamatay po yung tao or mamamatay yung halaman. So, kung nakikita po natin sa movies, yung namamatay sa cyanide poisoning, bumubula yung bibig. Kasi, kadalasan po ang lumalabas doon would be very, very concentrated cyanide together with carbon dioxide. Okay. Now, this would be for FADH naman. Now, the difference for FADH to electrons is that, if you remember, the breakdown again happens in complex 2. So, FADH2, dalawang electrons pa rin po ang na-form nito, pero ang mas mahina po ang FADH2 dahil ang electrons po niya, pa-isa-isa po lang niyang binibigay kay ubiquinone. Unlike for NADH, tigda-dalawa po talaga ang pasa niya ng electrons. So kung mahina itong mag-donate ng electrons at mas malakas mag-donate ng electrons si NADH, mas matindi ang contribution ni NADH sa pag-push ng hydrogen ions from matrix to intermembrane space. Okay? So ito po ang path ng FADH2 electrons. From 2, bibit-bitin ni ubiquinone, dadalhin sa 3. Magpapump ulit tayo ng 4 hydrogen ions from matrix palabas ng intermembrane space. Ang electrons po natin dito, bibit-bitin ni cytochrome C, dadalhin sa 4. Again, ganun po yun ulit. Dalawa lang ang ihihigop ulit dito na ilalabas dun sa intermembrane space. Bakit? Kasi gagamitin ulit natin yung hydrogen ions sa pag-form ng water. Ito pong water na ito is actually the metabolic water that we call. Kaya meron po yung span of time that we are able to survive even without drinking water. Because the breakdown of fats or proteins or sugars in our body would be bringing about water just the same. Pero syempre, to quench the thirst, this would not be sufficient enough. Okay? So I hope we are clear with that. Let's move to the result of the increase in hydrogen ions dito sa taas. So you could see that there are less hydrogen ions in the matrix. There are more hydrogen ions in the intermembrane space. Okay? The difference between the two spaces across the inner membrane is what we call as a gradient or pagkakaiba ng concentration. Here you have the higher hydrogen ion concentration. Here you have the lower hydrogen ion concentration. Now specifically, this is what we call as an electrochemical proton gradient. Okay? If you are familiar with the sodium-potassium pump of the nervous system, halos pareho po nito. Yun nga lang po, iba ang mga ions involved dito. Okay? So this would be higher. Now, the higher number of hydrogen ions here brings about a potential energy. Kumbaga, parang we would expect these ions in terms of their number kasi, they would, the natural flow would be from higher concentration to lower concentration. But if you remember, we mentioned that the inner membrane is impermeable. Wala pong nakakatagos dyan. So meron po silang dadaanan lang. Very specific lang po ang dadaanan nila. And this would be the ATP synthase. Now, chemiosmosis, chemiosmosis would be, in contrast to ETC, chemiosmosis would be bringing in hydrogen ions. Kaya ang arrow po natin nagtuturo pababa. ETC would be bringing out hydrogen ions at 3 po ang in charge doon. Which is why natural natataas dito kasi 3 ang nagko-contribute. Dito, isa lang ang pababa. And pa-isa-isa lang po ang pagbagsak ng ating hydrogen dito. How does this really occur? So we have the ATP synthase. This is the ATP synthase, which is also called as F0, F1 ATPase. There are different types of ATPases. Maraming iba-ibang klase yan. Pero this type or itong structure na to is the F0, F1 ATPase. So from the term synthase, it processes ATP. So this part here in red box is the F0 part. This part here down below is the F1 part. Okay? So balik tayo sa F0, eto. Ano po ba ang function nito? Now here you would see the hydrogen molecules to be flowing down. Okay? And then the hydrogen ions, to be flowing in, and the hydrogen ions to be flowing down. So here you see that as they move in from intermembrane space, then this turns. Puposisyon si hydrogen natin dito. Ito may existing hydrogen, ito may existing hydrogen. Bawat bagsak ng hydrogen natin dito, umiikot ang ating F0. You're familiar with toy cars na kinikwerdasan or relo na kinikwerdasan. That is similar to that because we are building mechanical energy. So there is mechanical energy that is built up here. So when all the spaces of the F0 is completely filled in with hydrogen ions, yung iba babagsak isa-isa, and then yung blank na space would again be filled up by another hydrogen ion. Okay? So parang dire-diretso siyang kumakwerdas, dire-diretso din ang ating build-up ng energy. Now what happens as that occurs? Down below here in our F1, our F1, habang nagbe-build up ng mechanical energy itong si F0, eto, nag-a-attract naman siya ng ADP at P. So bawat bagsak ng hydrogen ion dito, nagko-combine si ADP at P, forming ATP. Okay? So the mechanical energy in F0 would actually bring about the phosphorylation that happens in F1. Okay? So I hope okay na po tayo dyan. That actually concludes oxidative phosphorylation, which is electron transport chain, and chemiosmosis. This would also end cellular respiration. Thank you for listening po. Let us keep safe po pagdating sa ating COVID at saka sa ating init ng panahon. Thank you so much po.
Thank you very much, Ma'am Madge. We now open the floor for questions. If there are questions, LJ, you may unmute the microphone of the participants for those who'd like to throw in questions, or you can just write in the chat box your questions, and Ma'am Madge will read it and answer it for you. Anyone would like to start the question?
Ito po ang isa. From John Angelo Almazar, in the Krebs cycle, the reduction of carbon is it due to the release of carbon dioxide? There is no reduction of carbon. There is only a reduction in the number of the carbon groups that would be found in the molecule. Hindi po nire-reduce yung carbon. Yung molecule po ang binabawasan natin ng carbon. Kaya yung 6 carbon molecule po magiging 5 carbon molecule, tapos magiging 4 carbon molecule ulit. That's because of the release, the release of carbon dioxide po. And then, ma'am, what are the specific types of proteins po ulit for outer membrane and inner membrane? For the outer membrane po, we have porins. That's P-O-R-I-N, porine. These are water channels. Daanan lang po ng water yung mga yon. Kung minsan yung water po na dadaan, may kasama po siyang mga ions, pero hindi po yung ganong kalalaking mga ions. For the inner membrane po, ang proteins po natin doon, major protein natin is cardiolipin. That is C-A-R-D-I-O-L-I-P-I-N. And we also have the transport proteins. You remember the complex 1, 2, 3, 4, the prosthetic groups. These are still proteins. The ATP synthase would still be a protein. So lahat po yon kasama sa inner membrane. Does one glucose molecule always yield 38 ATPs at the end of ETC? If no, are there factors that would affect the production of ATP molecules at the end of ETC? For one, kasi, ang NADH forming ATP is not necessarily 3:1. It is usually from 3 point something. Hindi po siya eksaktong 3. Ganon din po sa FADH. Hindi din po eksaktong 2. Actually, sa NADH, it ranges from 2.5 to 3. Tapos sa FADH2 po, it ranges from 1.5 to 2. So po pwedeng mag-decrease po ito. Kaya po ang usual na range natin is 36 to 38 ATPs. Now, another is that, if you remember, yung NADH natin na galing sa glycolysis, po pwedeng hindi po siya pumasok agad dun sa ETC. You remember, it would still be free-floating from glycolysis. And then po pwedeng gamitin po ito ng ating cell or ng mitochondrion ng cell pag hindi po ito pumasok sa mitochondrion at po pwedeng gamitin po sa fermentation. Maalala niyo po yung fermentation natin, it would always break down NADH to NAD. Naalala po natin yon. So, yun po yung mga factors na kasama dyan. Okay. Ma'am, clarify ko lang po, is there any difference in the overall output of ATP between eukaryotic and prokaryotic organisms? Yes, definitely there is. Because for one, hindi po lahat is talagang from glycolysis to Krebs cycle to electron transport chain. Hindi po yun yung buong flow. Kumbaga po pwedeng nag-glycolysis, tapos nag-Krebs cycle, pero yung ibang NADH doon, hindi po necessarily pupunta agad sa ETC. Or parang yung binanggit natin kanina, na yung NADH sa glycolysis, hindi rin po siya automatic na papasok din po sa electron transport chain. So iba-iba po yan. Sa eukaryotic organisms, magkakaiba din po. Kasi ang cellular respiration, I really do not want to go into much detail muna, pero cellular respiration of plants is different from the cellular respiration of animals. Likewise, the cellular respiration of animals would be different from the cellular respiration of fungi, and other prokaryotic organisms like bacteria or algae. They would still be prokaryotic. So yes, iba po. What is the practical application of cellular respiration? Marami po siyang practical applications. Actually, dito po tayo nabubuhay. So, yung parang pagka sinasabi po natin yung mabilis ang metabolism ng isang tao, kaya hindi po siya mabilis tumaba, maski marami po siyang kinakain, isang application po yan ng cellular respiration. Ibig sabihin, mabilis pong mag-undergo yung buong process from glycolysis to Krebs cycle to electron transport chain. Hindi po napuputol dun sa fourth step. Maalala niyo po yung fourth step ng glycolysis natin? Hindi po siya napuputol sa fourth step or hindi po siya nagsuslow down sa fourth step. Yung iba po kasi magsuslow down sa fourth step, kaya nako-convert yung ating G3P into DHAP. If you remember, DHAP would be transformed into triglycerides and the triglycerides would be stored as fats. Kaya nagiging sexy po tayo dahil doon. Kaya thankful na rin po tayo doon. Other applications pa. Yung binanggit ko din po kanina na metabolic water, if you remember, that would be formed in the part of Krebs cycle and in the last part of the electron transport chain. So you have the formation of water there. Also, nabanggit ko din yung poisoning. May mga poisons po tayo na talaga ang tina-target is yung mga complexes po natin sa buong glycolysis, electron transport chain, Krebs cycle. May mga poisons po tayo na tina-target doon. So at one point in time na matatarget ang isang enzyme doon or ang isang structure doon, na-i-inhibit na po yung buong process no. How many ATPs are yielded in one glucose? Some reference book said 36, others 38. I think I was able to answer this already. May mga pagkakataon ba, ma'am, na nagkakaroon ng shortage of cellular ATP? Anong sakit kaya ang possible related to this? Matanong din kasi mga bata minsan, if that's so. Cellular ATPs, the only instance by which cellular ATPs, if I remember isa lang ang naiisip ko ngayon sorry. Pero the only instance that comes into my mind right now is during necrosis. If you remember, necrosis would be the death of a tissue. Ibig sabihin, namamatay yung mga tissues kasi walang oxygen na nagagamit. Ganon din po pagka yung sa ischemia or stroke, wala pong natatanggap na oxygen yung brain cells. So hindi po siya nakakagamit ng oxygen, hindi din po nag-undergo ng cellular respiration. So wala rin po siyang na-form na ATP. Wala pong gagamitin na energy yung cell. Yun po yung mga naiisip ko as of the moment, ma'am. What strategies to enhance ATP? The present strategies nowadays, eto na po yung mga makikita po natin sa mga article, aging, mga process na yan. Kasama po dun sa mga, di ba umiinom po tayo ng mga supplements like vitamin C, vitamin D na may kasamang magnesium, umiinom tayo ng may zinc, etc. etc. Isa po sa mga bagong supplements ngayon would be the NADH molecules that are placed in capsules. Okay? So siguro, yun yung isa na pagka syempre marami kang input ng NADH, mas ma-enhance yung pag-form mo ng ATPs. So pag mas marami ka naman ding energy sa cell, then mas madalas ang pag-renew ng cell parts mo. Mas madalas ang pag-renew ng cell parts mo, then, syempre, mas bago yung itsura ng cell, mas bago ang pag-function ng cell, then you are younger to how you, that is how they mention it. This is coming from a Harvard study. Are there signals for the cells to stop making ATP when they have enough? From what I remember, they really do not stop. They just slow down if there is too much. Pero they do not really stop. What happens is, pagka masyado ng marami yung ATP, they slow down in a sense that, if you again remember the split of the molecule from G3P and DHAP, yung split ng molecule po na yon, hindi na po siya magiging G3P. Siya po ay magiging DHAP. Tapos masustore na po siya. Magsuslow down therefore ang ATP production. Is ATP molecule the energy itself or just a carrier of energy? The ATP molecule is a molecule. Pero in the formation of the ATP molecule, it absorbs energy. So therefore, it is a carrier of energy. Kumbaga, para siyang pera, para siyang currency. Na kumbaga, yung piso, pag may bibilhin ka na worth na piso, hindi naman talaga yung worth kumbaga symbolism lang yung pera. Pero yung pera ang nagdadala ng kung magkano yung dala mo, kung magkano yung kaya mong i-trade. Nakuha po ba natin yun? I hope na answer ko po yun. Ma'am, di ba walang mitochondria ang prokaryotic cells? Saan sila gumagawa ng ATP? Siya po mismo yung mitochondria, kumbaga. Kasi dun po usually ang mga prokaryotic cells po natin, di ba meron po sila kadalasan dalawang membranes. Yung pong second membrane nila o yung inner membrane po nila, yun na po yung tadtad ng mga enzymes para po sa ATP production. Ang meron lang po talagang enclosed organelle dun sa ating prokaryotic cell would actually be the ribosomes. Not even for the DNA or RNA, kasi yung DNA and RNA would only be localized. Ma'am, meron ka po ba dyan simplified diagram ng cellular respiration, more especially for our students? Yeah, I have naman. I could share it to you. I-message nyo na lang din po ako para kung meron po akong maitutulong sa inyo, I'd be very willing to help po. All mitochondrial diseases po ba are genetically inherited? I'm not really sure, I'm not really sure. But in the evolution of the mitochondrion kasi, it is actually a very, very specific prokaryotic cell which has evolved to stay within a larger eukaryotic cell. So hindi po siya so far nag-evolve ng maraming beses. In more ways than one, we could call it as conserved. Which is why, medyo ang isang tawag natin sa mitochondrion or yung genes na dala ng mitochondrion is maternal genes. Di po ba? Kaya pwede po na lahat ng mitochondrial diseases are inherited. Pero I'm not so sure, again, may I just repeat that because marami po kasi ang advancement ngayon. Hindi po tayo usually maka-cope up sa sciences sa dami ng advancement. How about those in keto diet? Will there be a time for a short change of ATP? Wala naman po. Kasi kasi di ba we mentioned that glucose would be the start of the breakdown of glycolysis. However, pwede po na gumamit din tayo ng fats. If you remember, there is what we call as ketogenesis. So, meron din tayong ketosis. So, yung fats natin, natatransform po siya sa DHAP, dun po sa ating fourth step. Tapos, ay, sa fifth step, sorry. And then, matatransform po siya into G3P. And then, mag-undergo na po ulit siya ng buong cycle po ng cellular respiration. How respiration is different from photosynthesis. 1. The structures involved in respiration and photosynthesis. Respiration, by the way, teachers, iba po ang respiration from ventilation. Ay, saglit lang po ha. Medyo malulubat na po ako. Mag-charge lang po ako. Saksak ko lang po itong charger ko. So, respiration po, in terms of cellular respiration, ang in charge po dyan ay mitochondrion. Ang in charge naman din po sa photosynthesis ay chloroplast. Yun po ang isang difference. Pangalawang difference, ang respiration po is from large molecule to simple molecules. Catabolic process po siya. Ang photosynthesis po, baliktad. From simple molecules, papunta po ng large molecules. So, anabolic naman po siya. For the other details po, marami po actually differences ang respiration at saka photosynthesis. Pero, if given the chance to discuss photosynthesis, siguro that is where I would be emphasizing on the differences and likewise the similarities of respiration and photosynthesis. Pero as of the moment, yun yung medyo mas madaling intindihin coming from our discussion. Pero with regards to the other details, siguro banggitin natin yan sa susunod. Ma'am, question po, there is a selection from a book po kasi, ika-clarify ko lang po, eukaryotic and prokaryotic organisms differ in ATP output since po yung eukaryotic organisms po uses active transport of pyruvate patungong mitochondrion and prokaryotic po wala. Yes, actually. Kasama po yan sa mga differences. Pero, tulad dun sa question po kanina, kung meron po bang differences ang ATP output nila, eto na po yung isang difference. Okay. I think that's the last question po. Thank you very much po for your questions. If you have any other questions po or if you would like to contact me, you could just PM me po. I could help you out with anything that you would want to be helped out with. Thank you po.
Okay, thank you very much, Prof. Maj. Stay safe, people, and hope that you will continue to follow our SCI Science Teacher Academy for the Region's Facebook page for announcements of our future webinars. So, hello and goodbye, people. See you again in our future seminar.